which are simple, I read it and going fast, but if you find any problem or if you have question immediately, ask me, because today we want to finish this part at the earliest and we finish the remaining lectures. Conjugative pili, conjugative or sex pili are some of the most common bacterial pili. As the name suggests, these structures are involved in the transfer of genetic material from one cell to another. Compared to the other pili, the F. pilus or F. sex pilus encoded by F plasmid. The F. pilus is found in male gram-negative bacteria or F plus. So we have them in gram-negative bacteria, and those gram-negative bacteria with these types of pilus are known as male gram-negative bacteria. Based on electron microscope, these appendages have been shown to be dynamic and therefore elongate and refract continuously. With regard to the structure, the pilus is a polymer that consists of protein pilin and is also structurally similar to F-like pilus PED 208. Depending on the type of pili, the building blocks, proteins, may be arranged in a helical manner. 5 start helically filaments or in pentamer layers on top of each other. Given that bacterial pili originate from membrane, starting from membrane, they also consist of phospholipids and proteins. So mainly they contain protein, but some phospholipids are attached also, which form a protein phospholipid complex. However, the lipid in the pilus has been shown to be slightly different from the lipids in the cytoplasmic membrane. F. pili measure about 20 micrometer in length. Functions of conjugative sex pili. Conjugative pili are primarily involved in the transfer of DNA from one bacterial cell, male F plus, to another F minus. This is why they are also referred to as sex pili. While the process is not properly understood, it is suggested that single strand of DNA can pass through the hollow lumen of the pilus to be transported to recipient cell. The F. pilus is also thought to play an important role in the identification of recipient, as well as promoting contact between the two cells, the donor and recipient cells, as it retracts. Another pili is type 4 pili. Type 4 pili. These pili are a type of pili found in some gram-positive bacteria like Clostridia, and the majority of gram-negative bacteria. So it is mostly can be found in gram-negative bacteria and in some gram-positive, especially in Clostridia, are found. They have several important functions ranging from their role in locomotion to DNA exchange. So what is the similarity and difference between this pili with conjugative pili? Conjugative pili, previously we explained, was used only for DNA exchange. Transfer of DNA from plasmid from one bacterium donor cell to recipient cell. But here, along with conjugation, it can be used for locomotion. We had locomotion, we were talking about locomotion, about flagella, but here we can talk about locomotion or motility by type 4 pili also. That locomotion and motility by flagella are very fast. But locomotion by pili is very slow. Like conjugative pilus, type 4 pili are tube-like. So like conjugative, it is tube-like. That originate from the membrane. Yes, it starts from membrane. While they are strong structures, they are also flexible, which allows them to effectively perform their functions. Structurally, type 4 pili are also polymers that consists pilin. So it is also pili and it consists of pilin protein. Here, numerous copies of pilin, thousands copies, are polymerized from subunits in the membrane to construct the filament type 4 pilus. Although the polymerization of these pilin copies is not properly understood, it is generally believed to involve the assembly of ATPase as well as the core protein of the inner membrane. Pilins, subunits of pilins involved in the formation of type 4 pili are divided into two main categories that include type 4A pilins, type 4B pilins. So two types of pilins are come together for type 4 pilin formation. Type 4A pilins characterized by 5 to 7 amino acid signal peptides and phenylalanine. And type 4B pilins, which consist of relatively longer peptide and a hydrophobic residue. The pilus originating from the membrane to external and outwards into the outer environment. Type 4 polymers also have a helical orientation with a diameter of about 6 nanometer and 1 micrometer in length. Depending on the species, the pilus may be glycosylated, some sugars added, or phosphorylated, some phosphate added to the pili. Functions of type 4 pili: as the pilus is originating from the membrane to extend outwards into the other environment, type 4 polymers also have a helical orientation with a diameter of about 6 nanometer and 1 micrometer in length. So here you can see the image of flagellum, archeum, which it is related to archae, and type 4 pilus. Type 4 pilus. You know, we have 3 domains. 3 domains. Number 1 is eukaryotes. Second one is prokaryotes. And third one is archae. Groups of living things. All living things can be divided in 3 groups. Group 1, eukaryotes, like human, animals, protozoa, fungi, algae. Prokaryotes, like bacteria and archaea. In previous books and in previous times, some scientists believed that archaea are archaebacter and they are bacteria. But nowadays, it is obvious archaea are archaea. They are not eukaryote and they are not prokaryote. They are in separate domain. They are in separate domain. They have some similarity to prokaryotes. They have some similarity to eukaryotes. And they have many differences with prokaryotes and eukaryotes. Only 46% has similarity to prokaryotes and eukaryotes. And 54% there is not any similarity to prokaryotes and eukaryotes. But most similarity of archaea are related to eukaryotes, not to prokaryotes. So archaea, according to the biochemistry, according to the genetics and many aspects, look like eukaryotes and human than bacteria. So you can see the structure of flagellum in bacteria, archaea type 4, archaeo, something like pili, and type 4 pilus, located on the bacteria. This type 4 pilus can be used for motility. This type of motility is known as twitching motility. How is it? The pilus on the surface of the bacteria extending, type 4 pilus extending. B, the extending pilus come to contact with, adheres to the surface. The pilus retract. The pilus retract, pulling the bacterium to the direction of the arrow, red arrow. In this image, make the bacterium to go to right side. So this is a type of motility, not at the speed of motility by use of flagellum, but by the way, it is a snail-like motion. Slowly can have a movement for some purposes. So don't forget, type 4 pilus is used for motility as well as conjugation. A single bacterium may have several type 4 pili on its surface extending and retracting independently of each other. One of the usage of the type 4 pili is for biofilm formation, which last time, a few days back, we were focused on biofilm formation as a virulence factor in pathogenic bacteria. So biofilm formation was very important characteristics or virulence factor of bacterial pathogens. A biofilm refers to the aggregation of microorganisms which allows them to overcome stressful conditions. While type 4 pili are involved in motility, allowing the cells to move to given sites, they have also been shown to play an important role in the formation of microcolonies and consequently biofilm formation. Here, they not only adhere to the surface and cells, but also promote to close proximity between cells during biofilm formation. Some of the other functions associated with type 4 pili include electrical conductivity, adhesion of bacterial cells to eukaryotic cells. So it is very important. It is invasiveness. So our cells are eukaryotic cells. And bacteria, by help of type 4 pili, can attach to our cells, starting invasiveness is started by attachment by help of adhesins. And one type of important adhesin is type 4 pili. It is the type 4 pili. Protein production also is done. That, along with conjugation as well as motility, one another thing is related to type 4 pilus. You can see some bacteria outside are destroyed and some small pieces of their DNA are present in the environment. Type 4 like pilus is working to find and attach to these fragments of double-strand DNA and helping for transportation of these fragments to inside the cell for two purposes. In one purpose, the DNA fragments are rich of adenine, guanine, cytosine, thymine, as well as deoxyribose and phosphate, so they can be used for further replication. On the other hand, these small pieces of DNA may contain some genes, some good genes, which can incorporate to the DNA, to the chromosome of the bacteria. And that bacteria become stronger than before. So, number one, type 4 pili is used for conjugation. Number two is used for motility. Number three, for transfer DNA, foreigner DNA fragments from environment to the cytoplasm. Retraction or trap model showing key components of the DNA uptake system in Streptococcus pneumoniae, a long type 4-like transformation pilus is assembled on the surface of bacteria and directly bound to exogenous DNA and helping for transportation from outside to inside the cell. Type 5 pili. Type 5 pili is a unique type of pili found in bacterial species of the class Bacteria. While the mechanism through which this structure is assembled is not clearly understood, researchers have suggested that it involves protease-mediated polymerization. Like type 4 pilus, this pilus is also involved in adhesions. So it is a type of adhesion, as well as biofilm formation. You can see different subclasses of type 5 pili. Mostly they have many similarity with a few differences. You can see their structure. After pili, it is the time to talk about Fimbria. After pili, now it is the time for talking about Fimbria. Also known as attachment pili, sometimes we call Fimbria as attachment pili. Fimbria are shorter compared to pili and numerous in number, so match number and shorter than pili, ranging from 100 to 600 filaments per cell. Depending on the type of bacteria, Fimbria may be located at poles of the cell or evenly distributed over the surface of bacterial cell. Because they are shorter, Fimbria are stiffer compared to pili. There are several types of Fimbria, which include type 1 Fimbria. Type 1 Fimbria can be found on the surface of many gram-negative, example coliforms such as E. coli, where it is involved in adhesion. They range between 1 and 2 micrometer in length and about 7 nanometer in diameter, with, like all the other Fimbria, type 1 Fimbria are very stiff and don't bend much. Moreover, they have been shown to be composite structure consisting of a shorter and thin tip fibrilline located at the distal end of the rod. As is the case with pili, type 1 Fimbria are also characterized by helical orientation. In this case, the structure has a rigid-handed helical orientation consisting of 27 Fim-A subunit and about 8 helical turns. Type 1 Fimbria are also known as mannose-sensitive Fimbria or somatic Fimbria. So the other name of this fimbria is monos, sensitive fimbria, or somatic fimbria. Functions of type 1 fimbria. Functions of type 1 fimbria. Type 1 fimbria are some of the most common structures in the family of Enterobacteriaceae. So this type of fimbria is present mostly in Enterobacteriaceae, like E. coli. Here they are involved in adherence of bacteria to the cells of the host. However, this binding has been shown to be specific to glycoprotein that consists of one or N-linked high mannose structures. For this reason, type 1 fimbria are assumed to promote infection of the lower urinary tract and some mucosal surface. So because of the type 1 fimbria, we have urinary tract infection mostly by Enterobacteriaceae and by coliforms. Schematic representation of Salmonella type 1 fimbria. 1, the gene cluster related to this, and B, structure of fimbrium. This fimbrium, 1 fimbria. C, biogenesis by chaperone-Usher pathway. So the biosynthetic pathway of production of this fimbria is known to chaperone-Usher pathway. Type 3 fimbria. Type 3 fimbria are thin fimbria ranging about 5 nanometer in diameter and between 0.5 and 2 micrometer in length. Like type 2 fimbria, type 3 fimbria are especially common among numbers of family Enterobacteriaceae and especially to Klebsiella. Here, the subunits, building blocks, are first transported to the periplasm through the general secretory pathway, where a chaperone encoded by the gene MRKB promotes the development and assembly of the fimbria. Like pili and other fimbria, the subunit of type 3 fimbria are polymerized in a helical orientation. Beta strands in the C-terminal region of the subunits provide structural support to the appendage. Klebsiella pneumonia type 3 fimbria you can see in this image. Functions of type 3 fimbria. Type 3 fimbria play an important role in adhesion of bacteria to abiotic surfaces as well as formation of biofilm. For bacteria like Klebsiella pneumoniae, attachment to surface in catheters results in aggregation followed by biofilm formation. This is beneficial for the bacteria in that it promotes pathogenesis by promoting antibiotic resistance among some species of bacteria. Last fimbria is Curly fimbria. Curly fimbria. What is that? Curly is another type of fimbria found in gram-negative bacteria like Escherichia and Salmonella species. In Escherichia coli, biogenesis of the fimbria has been associated with at least 6 proteins, which are themselves encoded by CSG, PA, and CSG, DEFG operons. Like the other fimbria and pili, chorley fimbria also extend from the outer membrane. Functions: chorley fimbria are primarily involved in biofilm formation. For instance, using this fimbria, studies have shown the bacterium Salmonella enterotitis to adhere to the surface like stainless steel, and the subsequent formation of biofilm, which contaminate food material. Here you can see chorley biogenesis system of this type of fimbria. Differences between pili and fimbria. Differences between pili and fimbria. Both pili and fimbria are important filaments consisting of proteins that originate from the membrane and extend outwards. As such, they are visible under electron microscope because they are antigenic. The two filamentous appendages can also evoke an immune response in vivo. While they have a number of similarities, the two also have several differences that include: 1. Size. As mentioned, although they are both shorter and generally smaller compared to flagella, pili are longer than fimbria with hair-like appearance. Fimbria, on the other hand, are shorter with a bristle-like appearance. Bacteria. Although some gram-positive bacteria have been shown to possess pili, these structures are commonly found in gram-negative bacteria. Fimbria, on the other hand, can be found in both gram-positive and gram-negative bacteria, where they are involved in adhesion and biofilm formation. Distribution. Unlike pili, fimbria are numerous in number and tend to be eventually distributed on the surface of bacterial cells, whereas a single bacterial cell may contain between 200 and 400 fimbria on surface. The number of pili may range from less than 5 to 10 in total. 4. Function. While fimbria are primarily involved in attachment, which promotes biofilm formation, pili are involved in attachment, motility, as well as gene transfer from one bacterial cell to another. Fimbria have a more solid structure because they are primarily involved in attachment, while pili, especially sex pili, have a hollow tubular structure that allows for genetic material to be transported from one cell to the another one. So here in this table, you can see differences between pili and fimbria, sex pili and fimbria, long, thicker structure, thin and shorter present only in gram-negative bacteria, present both, less than 3 to 5 in bacterium, 300 to 400. Plasmid genes governs pili formation. Chromosomal genes in nucleoid governs fimbria. It works for cell attachment during bacterial conjugation, therefore they can be called sex pili. It works for surface attachment, and here you can see a conjugative pilus, which one strand of DNA is going to the recipient cell, and then by replication it will be double strand also. Another and final thing is capsule. Capsule. It is well-defined structure of polysaccharides surrounding a bacterial cell and external to the cell wall. So it is an outer layer of the bacteria. In most bacteria, except one, the structure of capsule is polysaccharide. But in Bacillus anthracis is an exception, because capsule of Bacillus anthracis is polydiglutamic acid. It is polypeptide, many amino acid of diglutamic acid. So only one capsule is protein, but the other capsules are polysaccharide. Slime layer. If it is so much compact, it is capsule, but if it is loosely come together, we call it slime layer. A slime layer refers to a loose network of polysaccharide fibrils that surround some bacterial cell walls. Observation is not possible by light microscope. After positive is possible. Observation of capsule is possible, but observation of slime layer is not possible because it is loose network. So by staining we cannot see slime layer, but by staining we can see the capsule. Is associated with adhesive properties of the bacterial cell, contain prominent antigenic sites. Functions of capsule. Functions of capsule. 1. Prevent the cell from desiccation and drying. Capsule or polysaccharide bond significant amount of water, making cell resistant to drying. Number 2. Attachment. Capsule helps in attachment on surface. For example, Streptococcus mutans that cause dental caries attached to teeth, surfaced by its capsule. Protection, it protects from mechanical injury, temperature, drying, and etc. Anti-phagocytic. Capsule resists phagocytosis by white blood cells. Capsule prevent attachment of bacteriophage on cell surface. Source of nutrition. Capsule is source of nutrition when nutrient supply is low in cell. Repulsion. Same charge capsulated bacteria repel each other. So here you see those bacteria which doesn't have capsule, the phagocytes, white blood cells, very easily can find them and then they phagocytosed. That those bacteria which contain capsule mask the surface antigens. So white blood cells and immune cells cannot find them. Glycoprotein molecules known as endocytic pattern recognition receptors are found on the surface of phagocytes. They are so named because they recognize and bind to the pathogen-associated molecular patterns. Components of common molecules such as peptidoglycan, teichoic acids, lipopolysaccharides, mannans, and glycans found in many microorganisms. Capsules can cover up these surface molecules, preventing their attachment to the endocytic pattern recognition sites on the phagocyte. Bacterial capsule preventing C3b receptors on phagocytes from binding to C3b are attached to a bacterial cell wall. So you can see the phagocytic white blood cells cannot find the antigens, surface antigens of bacteria, because they are covered by the capsule. And this is different types of adhesins present on different types of bacteria, and accordingly they can find their target cells and they can attach to them. And last page, we want to know the role of adhesins as virulence factors. We want to know the role of adhesins as virulence factors. One of the most important steps during pathogenesis of a bacterium is its attachment to mucosal or other biotic surfaces, such as respiratory, urinary, gastrointestinal tract. So start of infection is by attachment of bacteria to the target cells. The attachment to these surfaces in many bacteria has been attributed to the adhesins, thus suggesting their role as a virulence factor. In many cases, a bacterium can be rendered non-pathogenic by disrupting its attachment to specific tissues. However, the addition of adhesins to a bacterium doesn't convert it to pathogenic bacteria. The role of adhesins as a virulence factor was first shown in E. coli. The strains that attached to the intestinal mucosa of the pigs were disrupting the function of adhesins was able to convert the strain to non-pathogenic. Studies also shown that adhesins are potent virulence factors in specific strains of E. coli, pyelonephric strains that are present in human kidney. Okay, so our lecture over. We don't have anything else to explain. It is sufficient. So till this part, we have examination. If you accept, half an hour we have a rest, and then we will come back to explain about examination, and we terminate our course. So would you please at 6 p.m. again come back for a short time. We will talk about the examination, and if you have any scientific question, if you need any review, we will go through it. So this lecture over. We will meet each other after a short time at 6 p.m. again. Thank you very much. I'm waiting to meet you again. Goodbye.